Alright guys, welcome back to Underground Science, and in this video we're going to be going over the interactions between a macrophage, B-lymphocyte, and a CD4 positive cell. Alright, so let's go and get started. So if you guys remember from our part 1 and part 2 video of our interactions between a B-lymphocyte and CD4 positive cell, right, we went through the fact that if we did a quick review of it, right, just to refresh our memory, so let me go and pull up a screenshot of that picture. All right, here you go. And so we went, oh crap, sorry about that. Let me go and make that a little smaller. All right, so we went over the fact that um, our B lymphocyte, so our B lymphocyte can't really start proliferating into more B lymphocytes, into more effector and memory B lymphocytes with the same memory bound antibodies if we don't have a helper T cell to bind to the specific antigen onto the B lymphocyte. So we said that, and we also said the CD4 positive cell is also known as a helper T cell because it has that CD4 glycoprotein on it. All right, so our CD4 is actually a glycoprotein. So it's made up of some carbs and protein. All right, so glycoprotein. And so we said that our CD4 glycoprotein has to bind to the MHC2 itself, and then our T cell receptor has to match with our antigen binding spot, right? So it has to be, so it's a specific, right? It's part of our specific immune system. So it really has to match the antigen. If it does, it'll bind, the CD4 will bind with the MHC2, then lymphokines and cytokines will be released, and these cytokines and lymphokines will trigger the B lymphocyte to start proliferating into memory B cells, and we said into plasma B cells, right? So memory B cells and plasma B cells. And then we said that we went on and said that in the part 2 version, I believe, is that what happens, not only does a helper T cell cause or help the B lymphocyte to proliferate into our memory B cells and plasma B cells, we said that our B lymphocytes can also release some cytokines. And what can happen is that these cytokines that the B lymphocytes release, we said that can trigger the helper T cells to start proliferating into generally two types of helper T cells. So it can start proliferating into effector helper T cells, right? And we said it can start proliferating into memory helper T cells, right? And I mean, it's the same for B lymphocytes, right? Memory B cells and effector B cells. So plasma B cells are the same thing as effector B cells, all right? So it's going to start proliferating to effector T cells and memory T cells. All right, so we're just doing a quick review of part 1 and 2 so we can, so when we talk about the interaction between macrophages also, it'll make more sense. Okay, so the cytokines released by the B lymphocytes can trigger proliferation of the helper T cells, and it can proliferate into effector helper T cells and memory T cells, or sorry about that, memory T helper cells. All right, and so that's what we said can happen, and then we said that these effector helper T cells, as a group, can start to release specific types of cytokines and lymphokines, and one type of cytokine we said that's really important is called interleukin-2, which we said can then further stimulate the proliferation of nearby T lymphocytes into cytotoxic T cells, and memory helper T cells. So memory helper T cells. So interleukin-2 is actually really important, okay? So knowing this, let's actually look at the interaction between macrophages and helper T cells, are also called CD4 positive cells, and B lymphocytes. All right, so if we look at a macrophage here, right, so let's go to another page. So if we look at a macrophage here, let's say we draw a whole huge macrophage, right? So we draw a huge macrophage. So let's go and label this as a macrophage. And let's say we have a MHC2 complex on it, and the macrophage, just to save time, we know that based on our antigen presentation video, we know that our macrophages can phagocytose antigen or pathogens and break it apart and antigen present it, right? So let's say we already have an antigen here that our macrophage is presenting on its MHC2 complex. All right, so remember this is a macrophage now, not a B lymphocyte. So what can happen is that if we have a T cell floating around here, right, and it's going to be like, let's say, it's not going to be active, okay? So we're going to have, let's say, a T cell, right? So we're going to have a T cell that's going to be floating around here, and it's going to be looking for a specific antigen binding spot that matches its to the T cell's receptor. All right, so this is going to be our T cell receptor here. T cell receptor. All right, sorry about that. T cell receptor. All right, and so what's going to happen is that this helper T cell, so it's going to be our helper T cell, is going to be floating around, and it's actually going to be inactive. All right, so it's not going to be active, because to be active, it actually needs to combine with a specific type of cytokine, right, or many cytokines, and it has to bind to something to be active. It's looking for something to bind. It's looking for a specific antigen that can actually fit its T cell receptor so it can bind onto it and become active. So then what can happen is that a next scenario that we can look at, let's just draw it a little smaller so we can stay on the same page, is that we're going to have our MHC2, right? And we're going to have our antigen here that our macrophage is presenting, okay? And then a next scenario can be, oh, it matches, right? A next scenario can be the fact that our T cell receptor comes over here and it matches. This antigen binding spot matches exactly with its T cell receptor. So it could look something like this. So it matches perfectly, right, with its T cell receptor. And so this is still our helper T cell. All right, and this is still our macrophage. Just going to keep on labeling these so we can stay on the same page. And this is our MHC2, all right, in brown, this one right here. Okay, so a match, right? But remember, we have another component to our helper T cell, which is why it's also called a CD4 positive cell, right? So the name actually gives it away, is that we have a CD4 glycoprotein that latches itself on to the MHC2 complex. We need both components, all right, in order for this activation process to occur. So let's go and color this in, right? And let's go and label this as our CD4 glycoprotein. All right, that's present on our helper T cell's membrane. Now, once the T cell receptor, crap, gotta label that too. Sorry about that. So this is our T cell receptor. All right. So once our T cell receptor binds onto the specific antigen binding spot, the specific complementary antigen, and then once our CD4 glycoprotein binds to the MHC2, what can happen is that our macrophage can release cytokines, but a specific cytokine is what we have to focus on, is called interleukin-1. All right, so this will then release interleukin-1. All right, so interleukin-1. So let's just draw a couple of circles here showing interleukin-1 release. And so what this interleukin-1 can do, right, so let's label this as interleukin-1. Right, so hopefully this video is not too long. All right, so interleukin-1 will focus on two main things that it can do. So first thing it'll do is it'll actually help activate the inactive, all right, it'll activate the inactive helper T cell. Almost wrote CD4 positive cell there, but it's fine. The helper T cell. And another thing it's going to do is that it can actually help in interleukin-1 can actually help in attracting, it can help attract, I should say, more white blood cells to the macrophage. All right, to the macrophage. So more white blood cells means more help to take care of whatever pathogen that we need to take care of. All right, so in this case, it's not macrophages, it's just one macrophage. All right, so that's two main things that we'll focus on that our interleukin-1 can do. All right, so let's go and draw that in. Okay, so let's go and show that, show how interleukin-1 activates the helper T cells. We'll come around here, right, and it'll come on the helper T cell and bind to the helper T cell, and this will induce the activation of the helper T cell. So now the helper T cell can be activated, and once it's activated, notice that now our helper T cell knows the specific antigen that it needs to go find. So once this helper T cell, so let's go and draw an arrow all the way over here. Sorry if it gets too messy, but now we have the activated version of the helper T cell, right? Ready to go, go beast mode, right? So it's going to have our T cell receptor that's fully activated. And so let's go and label. Now this is our active, this is our active, I don't know why I wrote it like that, but active helper T cell. All right, and from here, we'll see in the next video how it can go now. We can connect our previous video before this of the interaction between B lymphocytes and CD4 positive cells. We'll see how this active helper T cell can then go and find a B lymphocyte and induce proliferation there. All right, so see you in the next video.